Hi guys, welcome to my channel. My name is Greg Friedman, and here I review scientific papers. Call it the Journal Club. Matter of fact, I think starting today I will. I miss it from grad school, and now I'm doing it, well, with you guys. So thanks for joining me. Very interesting paper today. The concept is very scary for me lately. It's outside of my technical expertise, but I had a surgery some years ago, and one of the considerations was, am I going to make it? You know, antibiotics. There are bacteria in the hospital that resist them. What happens when there are no antibiotics left? Well, there's an alternative, which I've been looking into recently. It's quite intriguing to me. So let's dive right into this. There's a lot of work that was done in phage therapy. This was happening during the Second World War and before that. There's a lot of interesting history, which I will try to dig up for you one of these days. Maybe I'll find a paper on that. But today, we're looking at a review published by the American Society for Microbiology in the Journal of Antimicrobial Agents and Chemotherapy. And it is on considerations for the use of phage therapy in clinical practice. Bunch of authors. I didn't count them. A, B, C, D, E, F, G, H, I, J, blah, blah. Many. Like 15 of them. And look at that. Mayo Clinic, Albany College of Farm Blood, Johns Hopkins, NIH, UC San Diego, Duke University, Yale. I mean, big schools. Anyway, so increasing antimicrobial resistance and medical device-related infections have led to a renewed interest in phage therapy as an alternative or adjunct to conventional antimicrobials. Yes, that's a good idea. So conventional antimicrobials, we hear about, MRSA, methicillin-resistant Staph aureus, the other superbugs, Cinetobacter. This paper will talk about them, the so-called escape pathogens. There's a bunch of them, and they resist antibiotics. So what do we do when we don't have antibiotics? Well, somebody's gonna, like, billion-dollar pharma company, right? I don't think that's happening. But anyway, it's not what the paper is about. It's about phages. So the Antimicrobial Resistance Leadership Group has convened a panel of experts in phage therapy, clinical microbiology, etc. And they came up with this paper funded by the National Institutes of Health. It's a pretty big deal. All right. Antimicrobial resistance and increasing use of implantable devices, which are prone to biofilm-mediated infections, have contributed to decreased effectiveness of antibiotics. Among other things, patients may be left in a chronically infected indwelling device. So many patients. This paper doesn't give you numbers, and I'm not sure you care about numbers. If you have a loved one with an infection, phages might be a solution, so you should download this paper and read it. Of course, I'll leave a link down in the description. In late 2020, National Institute of Allergy and Infectious Diseases, NIAID, and the Antibacterial Resistance Leadership Group, funded by NIAID, convened a task force comprised of experts, and this is the output. These recommendations are not the position of NIH or NIAID, but you should read them anyway. So the task force was looking into the escape pathogens. I'm going to mess up reading these names. So what is this? Don't know what this guy is? Probably fecal bacteria. Strep? Acinetobacter, I know. Nasty bug. Pseudomonas and Enterobacter species. These guys, along with resistant gram-positives, were prioritized in this review. It's pretty long. So these guys tasked themselves with providing an evidence-based evaluation of circumstances where this experimental therapy might be considered, acknowledging that at this point no recommendation can be made. So they're not recommending anything, they're simply reviewing literature to check the current status. And they're asking themselves a bunch of questions. I think it's a dozen. First one, for which infections can phage therapy be considered? Well, there's a bunch of them that resist conventional antibiotics. So bacteriophage therapy is a consideration for bacterial, but not fungal, viral, or parasitic. Yes, bacteriophages are really, really tiny viruses that infect bacteria. Now, occasionally I heard some resistance to bacteriophages and they may evolve and infect humans. I don't think so. Viruses that infect bacteria, there's such an enormous distance between us and bacteria, genetically speaking, that I don't think it's easy for these guys to evolve to infect us. Who the hell knows? Anyway, so table 1 summarizes published clinical experiences from case reports. If you're concerned, if you have a multi-drug resistant infection, you should download this paper and look at this table. It's a little bit big, a couple of hundred references in this review. So you should study it if you're concerned. If you're just listening to this for sake of listening to it, well, you can come back to it later if you have a question. Great, great paper. They looked at recurring urinary tract infections, chronic rhinosinusitis and otitis media, skin and soft tissue infections, respiratory infections, bone and joint infections, PJIs, didn't know what that is, prosthetic joint infections. Yep, cardiac device associated infections. Sounds nasty. Sepsis. And always they mention gaps in the knowledge in the answers to these questions. So here, there's a real near total lack of randomized controlled trials of phage therapy. Near total lack of randomized controlled clinical trials. So that's kind of a problem. It's difficult. There are millions of phages. They're all around us. Probably most of the species haven't been discovered yet. So it's a biologic, but it's a challenge. And also we kind of want to analyze and definitely publish studies with negative outcomes. From the little reading that I've done on the topic, I haven't seen many negative reports. Does it mean phages are the ultimate solution? I'm not entirely sure. I'm reading this along with you. Second question, should antibiotics be administered concurrently with phages? Suggestion, the Taskforce suggests that if phage therapy is used, it should be in conjunction with conventional antibiotics. Okay. And the rationale for that is the recent clinical data on phage therapy generated primarily in compassionate use settings in conjunction with antibiotic therapy. See a bunch of references. Again, look, if you're considering this for yourself or someone you know, you should spend more than 10 minutes on YouTube. Just, personal suggestion. Of course, gaps in the knowledge. There is paucity of controlled clinical trial data. I had to look up this word, paucity. I love it. It is presence of very little. So there is a presence of very little of controlled clinical trial data. I'm going to use this word, paucity. It's awesome. It's like I have it, but I have very little of it, right? So yeah, definitely more clinical data is needed. Question 3, is phage therapy safe for clinical use as an anti-infective? The Taskforce determined that phage therapy is generally safe to administer with adverse events rarely reported. Well, that's good news. And again, if you're considering it, read this in detail, what the adverse effects were, if any. Question 4, what are some practical considerations when considering phages as an anti-infective therapy? Well, the task force suggests a detailed plan outlining uncertain clinical outcomes, lack of proven efficacy, lack of standardized dosing or administration, potential adverse events, costs, and other logistics be discussed with patient. So they didn't say anything. I think if you're considering this, spend a couple of days reading it. The infection isn't going to run away from you. Question 5. Which regulations govern use of phages in clinical setting? Oh, that's a big one. Taskforce suggests that compassionate use is a viable regulatory pathway for treatment of individual patients with phage therapy. My understanding of compassionate use is that's like a last line of resort. Like they tried everything else. Thousands of dollars in antibiotics. Days, if not weeks, in a hospital, and only then use phages. It's a biologic. Phages have been fighting bacteria for ever since. I don't know who was here first. So I think we might consider phages not as the last line of defense, perhaps as the first line, just an idea. And Taskforce also endorses concept of common database. It's tricky because, different strains of bacteria in Egypt and in whatever, Moldova and in Texas, they're going to be a little different. So common database, not sure about it. Maybe a bunch of different common databases. Blah, blah, blah. You see lots of text that I'm skipping. I read it, but I don't see the point of reading it all to you. If you're interested, paper is in the description. Understand which conditions should phage susceptibility testing be used to select phages for therapeutic use. While it would be ideal to perform phage susceptibility testing before phage administration, all right, what they're talking about is, do we make sure that the phage cocktail we're giving you is going to work on your bacteria? And they're saying, it'd be nice if there was a method, if it didn't take too long, hint, something, could be in development. Look into companies that are working on it. Maybe that's a worthwhile investment. Yep, the gaps in the knowledge, there's lack of standardized clinically available methods. It's early. It's just beginning. Antibiotics are on their way out. Phages are on their way in, but it's going to take some time and many, many, many billions of dollars to get it to work. Which parameters should the emerging phage susceptibility testing platforms consider? The task force couldn't identify a gold standard method. Methods that demonstrate lack of in vitro outside of the body phage activity against the targeted bacterium should be shown to correlate with unlikely in vivo activity. So they're saying they don't have a gold standard. They didn't find one. And if there is one, the test should correlate, the outside of the body test should be closely linked to the inside of the body one. That's more of a hint to whoever's trying to develop this. And again, gaps in the knowledge is there's need for standardization, reproducibility, speed. This needs to be fast. You don't want the patient waiting for 2 weeks. Blah, blah, blah. Lots of work to be done. Next question, number 8. Which parameters should test methods for assessment of phage activity in combination with antibiotics consider? Which parameters should test methods consider? Got it. The task force didn't find any standard methods. There's very little research in this direction. That should be fixed. This seems like such an amazing solution to the antibiotic-resistant problem that I think more research dollars should be spent on this by everybody in the world, not just United States. And they're also suggesting specific phage antibiotic combinations. So in fact, since the panel is suggesting continuing use of antibiotics as you're administering phage therapy, they're saying that should also be researched. Okay, that's a good suggestion, I think. Again, not really an expert, just reading it. I read this a few hours before I'm reading it to you. Next question, which immune system components are likely to impact safety and efficacy of phage therapy, and how can these be tested? Taskforce is unable to recommend specific assessment of immunologic parameters that correlate with phage activity that should be assessed due to a significant knowledge gap in this area. You have an immune system. You have bacteria invading your body, avoiding your immune system, and resisting antibiotics that are pumped in. So adding phages will somehow maybe begin lysing, popping bacteria open. And what's going to happen to your immune system? They don't know. I don't know. I wonder. I would imagine this would be, in adverse effects kind of a thing. And again, if you're considering something like this, read this in detail. It's a good review. Next question. What are current acceptable standards needed for safe phage administration? Taskforce suggests that phages used for phage therapy should not encode antibiotic resistance or toxin genes in their genomes and should not be capable of undergoing lysogeny. I have to look up what lysogeny is. Lysogeny, type of life cycle that takes place when bacteriophage infects certain types of bacteria. In this process, the genome of the bacteriophage steadily integrates into the chromosome of the host bacterium and replicates in concert with it. So, not sure if I understand the task force's suggestion correctly. They want phages that should not encode antibiotic resistance. Yes. Should not encode toxin genes. So they want phages that do not produce more antibiotic resistance or more toxins spilling into your body. I guess that makes sense. This is a little bit difficult one for me, so we'll skip it. I can do that. I'm in charge of the video camera. Gaps in the knowledge for that question. Computational tools. It's difficult to develop things where knowledge is pretty limited as it is currently. Next question. Under which conditions should phage be quantified in clinical specimens, and which parameters might be important features of assays to quantify phage in clinical specimens? Given the uncertainty as to the number of phages required at the infection site for maximal effect, the task force was unable to make a recommendation as to the circumstances under which phage concentrations should be measured in clinical samples. They don't know. I don't know. Phage is a virus that infects bacteria and makes a bunch of copies of itself. So theoretically, one's enough. Practically, that may not be the case. Do you need 2 million, 2 trillion? I don't know. And they seem to not know. So again, that's an unknown that will be solved with more research and clinical trials and all of that. And next question, what types of phage products are available and which are preferred for treatment of patients with acute and chronic bacterial infections? Suggestion is phages have been used as monophage and in cocktails, pre-biomanufactured, so biomanufactured in advance, or biomanufactured for the patient. In addition, both natural and bioengineered phages have been used. There's too much data to answer this question. So what types of phage products are available and which are preferred? They're saying, well, there's a whole bunch of different stuff. I read this suggestion as kind of no suggestion. I like the gaps in the knowledge. Optimal phage products for empirical and personalized use against groups of common bacteria pathogens should be developed and evaluated, preferably in well-designed controlled clinical trials. So somebody should spend a couple of $100 million to answer this question. I agree, actually. For, next time I need a surgery, I would like to take a phage cocktail that works for that particular hospital. That'd be awesome. What are the key considerations when selecting initial phage doses? Taskforce suggests using the highest safe and tolerated dose of phage product with endotoxin levels below the acceptable limit set by the FDA. So I think they're saying use as much as you can. If it's going to work, if it's going to work and it's not very toxic, just take a bunch of it because we don't know how much you need. That was a couple of questions ago, I think. Next question, what are potential routes of administration for phage therapy and how should they be selected? The best methods for administering phage therapy, I guess, has not been established. Yep, it's on case-by-case basis and hopefully your doctor knows what it's doing. Or she. Yeah, I like this. Inhaled administration has been used clinically for treatment of patients with respiratory tract infections, but the ability of inhaled phages to concentrate in anatomic components of the respiratory tract has not been systematically characterized in humans. Yeah, too little research in this area. Next question, 15 at this point. What dosing frequency and duration for phage therapy should be used? There are insufficient data. We know it works. It mostly works. Check the references, but they're all over the place. We don't know how much to give. We don't know when to give it, how to give it, and what to combine it with. Cool, but it works. It's pretty awesome. Finally, conclusion. Phage therapy has reemerged as a potential treatment for refractory infections in recent decades due to the increasing need for alternative or adjunctive anti-infectives to conventional antibiotics. It's hard to read. Yeah, it's coming. It's back. It was there before antibiotics. Antibiotics, hallelujah, but now they're failing, so we need to bring phages back. Totally. Although phage therapy is currently reserved as salvage therapy, ultimately the hope is that phages, if shown to be beneficial, will be able to be utilized earlier during infection, perhaps reducing upfront use of antibiotics, helping to preserve them. Or eliminate them completely. That'd be awesome. Some antibiotics are pretty toxic, especially the ones, the last line of defense ones. And of course, the authors acknowledge funding from NIH for this work. Amazing work. Look, there is a bunch of references. I'm still scrolling. 215. So wonderful paper. Again, if you're considering this therapy for yourself, your loved one, or a friend, send them this paper at the very least. Discuss this with a medical professional. Don't listen to a talking head scientist on YouTube. This is just my journal club. I read this paper and, reported it to you guys. I hope you enjoyed it. Please do, like and click the little bell icon. And I'll see you on my next, what am I calling this? On my next journal club. That's it. All right. Bye, guys.